My dear boys and girls, this is MD Rejaul Islam. Today it is the second lecture from chapter 1, Animal Diversity and Classification. So dear students, let's start. Today we want to discuss some other basis of classification of the animal kingdom, and first of all cleavage. What is cleavage? In a simple sentence, the repeated mitosis cell division of zygote up to morula stage, it is called cleavage. And in another way, we can also say the division of zygote, it is called cleavage. So look at here, cleavage is a process of mitotic cell division through which a unicellular zygote of sexually reproductive animal transform into multicellular embryo, it is called cleavage. The different cells derived from cleavage are called blastomere. Mode of cleavage is determined by the divisional plane of the zygote, distribution and amount of yolk in egg, and the fate of germ layer. On the basis of divisional plane of zygote, following three types of cleavage seen in the animal kingdom. First of all, radial cleavage. Secondly, it is bilateral cleavage, and thirdly, it is spiral cleavage. So look, first of all, radial cleavage. Radial cleavage defined as the cleavage where the embryonic cell or blastomere arranged in a radial symmetry around the divisional plane, like the radial symmetry. And this type of cleavage is found in the animals, in the zygote of the animals belongs to phylum Arthropoda. Bilateral cleavage, the first cleavage results in bisection of zygote into left and right halves. The following cleavage planes are centered on this axis and result in two halves being mirror image of one another that results in a bilaterally symmetrical embryo. It is called bilateral cleavage and it is found in the zygote of the animals belongs to phylum Chordata. Spiral cleavage, in this cleavage the first two divisions of zygote are oriented in planes that occur at right angles parallel to animal, that is vegetal axis of the zygote. At third division, the early micromeres, the uppermost smaller cells, rotate with respect to the macromeres so that the micromeres lie opposite to the furrows between the macromeres. The lowermost largest cellular layer, this is the macromeres, forming a spiral arrangement of the blastomere. And the example it is occurs in the zygote of the animals belongs to phylum Annelida and Mollusca. The next basis of classification, very important classification basis, it is the coelom. First of all, what is coelom? The liquid filled cavity between the alimentary canal and the body wall of multicellular triploblastic animal develops from mesodermal layer during embryonic development and bounded by peritoneal membrane is known as coelom. According to Hyman, coelom is the hollow space between alimentary canal and the body wall. The free space that present between the alimentary canal and the body wall, it is called the coelom, which is lined by a covering membrane. It is the peritoneum membrane, and the peritoneum membrane is derived from mesodermal embryonic germ layer. The animals are of three types based on the presence or absence of coelom. First of all, acoelomate animals. In these animals, coelom is totally absent, body cavity filled with mesenchyme and muscles. Example, it is Platyhelminthes, Tenia solium, Fasciola hepatica, and Nemertea. Pseudocoelomate animals, in these animals, the body cavity is not lined by mesodermal peritoneal membrane. That means body cavity is present, but the covering membrane is absent. It is called pseudocoelomate animal. Example, Nematoda, Ascaris lumbricoids, and Ancylostoma duodenale, Acanthocephala, Rotifera, Entoprocta, etc. Then eucoelomate animals, in these animals, true coelom is lined by mesodermal peritoneal membrane. That means coelom is present and the coelom is covered by peritoneum membrane. It is called eucoelomate. And the example of eucoelomate, it is mollusca, annelida, echinodermata, and chordata. The next basis of classification, it is about the notochord. Notochord is a flexible rod of large vacuolated cells derived from the mesodermal embryonic germ layer packed within a firm sheath found at some stage during the life of all chordates between the central nervous system and the alimentary canal or the gut. On the basis of presence or absence of notochord, animals are classified into two groups. Non-chordate, that doesn't possess any notochord, and the example it is phylum Porifera to Echinodermata. Chordates, that means the animals belongs to phylum Chordata. This is the classification of animals on the basis of notochord. That means on the basis of notochord, animals are classified into two types: non-chordate, who doesn't possess any notochord; chordates, who possess the notochord. The next basis of classification, it is polarity. The polarization of the body parts of an animal on the basis of mouth and head position is called polarity. The end of the body which bears the head is called anterior end, and the opposite end of anterior end, it is called posterior end. Similarly, the end with mouth is oral end and opposite is aboral end. This is the classification of animal on the basis of polarity. Then the next basis, it is tagmatization. What is tagmatization? Tagmatization is the specialization of metameres, that is the body segments in animals, particularly arthropods, into distinct body region specialized for specific functions. That means the specialization of the body segments into head, thorax, and abdomen of the animals belongs to phylum Arthropoda. It is called tagmatization. Then each segment, it is known as tagma. It plays a role in arthropods classification, where the body is divided into three segments: head, thorax, and abdomen. Look at here, this figure indicates the division of the body, the segments of the body. This is the head part, this is the thorax part, and this is the abdominal part. We can also discuss about the polarity. This is the head part, this part it is called anterior part. This is the opposite side of the head part, this is called posterior part. Here it is the mouth opening is present, this is oral part, and here it is the anus, that is opposite of the mouth, it is called aboral side. So this figure indicates both the tagmatization and polarity of animals. The next basis of classification, it is the classification of animal kingdom on the basis of presence and absence of alimentary canal. So on the basis of presence and absence of alimentary canal, the animals are classified into two groups. These are parazoa and enterozoa. What is parazoa? Animals in which alimentary canal is absent, it is called parazoa. And the example of parazoa, it is the animals belongs to phylum Porifera. And enterozoa, what is enterozoa? Animals in which alimentary canal is present, it is called enterozoa. And here it is, look at here, enteron means digestion. So which animals are enterozoic, that is belongs to enterozoa, the animals in which alimentary canal is present. And the example of Enterozoa, it is from Nidiria to Chordata. Then here it is some informations about the appendages. First of all, what is appendages? The locomotory organs of animals are called appendages. The organs through which an animal locomote, that organ or that organs are called appendages. The name of different locomotory organs or appendages found in the animal is mentioned in the following table. Look at here, the appendages, for example, nidoblast, tentacle, pedal disc. It is found in phylum Nidoria. By the help of nidoblast, tentacle, and pedal disc, a Nidorian animal, it changes its position. In case of Annelida, the locomotory organs, that is the appendages, are parapodia or seta. In case of Arthropoda, the appendages are joint appendages, wings, tailson, etc. In case of mollusca, the appendages are, that is in case of mollusca, muscular foot, it acts as appendages. In case of Echinodermata, tube feet, it helps in locomotion. And in case of chordata, different types of appendages like fins, wings, limbs, etc. So these are the list of appendages that found in different animals of the animal kingdom of different phyla. So after completing lecture 2, some homework for you. First of all, draw the level diagram of different symmetry. Draw the level diagram of different coelom that found in the alimentary canal, and draw the level diagram of notochord. So dear boys and girls, this is all for today. Be fine everybody, and thank you so very much.